Okay, let's dive right into this explainer and uncover the highly coordinated espionage and military operations happening inside your body literally right now. You might think of your immune system as just a basic shield, but honestly, it's actually a dynamic, ruthless military force operating in constant warfare against microscopic invaders. So, to really understand our body's defense, we've got to first ask, how does it identify an invisible enemy and orchestrate a massive systemic counterattack? The moment a pathogen, like a virus or bacteria, breaches your physical barriers, well, the clock starts ticking. But your body doesn't just blindly fire into the dark, right? It needs intel, it needs communication, and it needs a super precise strategy to neutralize the threat before it takes over your vital systems. All right, meet your frontline defenders, organized into these really distinct tactical units. First up, we've got the innate immune system. Think of neutrophils as your rapid response street cops. They're the first on the scene, quickly dealing with immediate threats. But the real magic, that happens at the communication bridge. The dendritic cell acts as this incredible intel-gathering signalman. Its entire job is to collect evidence from the battlefield and relay it to your lymphocytes. And these B and T cells, they are your elite special forces, trained for a highly specific, targeted response. So the crucial point here is understanding the enemy's intel. What exactly are these signalmen collecting? Well, they're looking for an antigen. And an antigen is basically any foreign substance, whether that's the invading pathogen itself, a toxin, or a viral particle that induces an immune response in the body. But here's the thing, the special forces don't just look at the whole invader, they're looking for the epitope. The epitope is a highly specific, microscopic fragment of the antigen. Think of it as the enemy's unique fingerprint. It's the exact, precise molecular shape that our forces are scanning for to confirm a target. Now, the dendritic cell is absolutely your ultimate biological spy. Starting out as an immature cell just patrolling the tissues, it actually swallows up viral proteins as soon as it encounters the enemy. Once inside, it breaks the enemy down into those crucial epitopes we just talked about. And as it processes this intel, the dendritic cell physically changes shape. It extends these long star-like arms to carry this sort of molecular wanted poster away from the front line, also the special forces can read it. From there, our spy travels through the lymphatic system. And this isn't just some random assortment of tubes. It's a sprawling, dedicated highway network connecting every single battlefield in your body to hidden command centers. Riding the current, which is actually moved only by the movement of your own muscles, since this system doesn't have a pump like your heart, the dendritic cell completely bypasses the normal bloodstream. This ensures its highly classified intel gets directly to the generals of your immune system without any chance of interception. And those hidden command centers, those are your lymph nodes. If you've ever felt swollen glands in your neck during a cold, you're actually feeling this bustling military base in action. Our spy enters through the afferent lymphatic vessel and moves right into the T cell area. It essentially stands on a soapbox, presenting the viral intel to thousands of circulating, naive special forces, just waiting until it finds the exact T cell that matches that specific viral fingerprint. Depending on the intel presented in the lymph node, the command center is going to launch one of two massive strikes. First, we have the humoral response, driven by B cells. You can kind of think of this as the artillery. They're launching targeted antibody missiles from afar to neutralize viruses just floating around in your fluids. On the flip side, we have the cell-mediated response. This one is driven by T cells, which act as the infantry. They engage in direct hand-to-hand combat to seek out and destroy your own cells that have already been compromised by the virus. It's honestly mind-blowing to think about the sheer scale of the deployment as your body diverges its resources here. In the humoral response, B cells transform into plasma cells, becoming literal antibody factories that rain down Y-shaped proteins to bind to the virus. Meanwhile, in the cell-mediated response, helper T cells are firing off chemical flare guns called cytokines. These flares command cytotoxic T lymphocytes to mobilize and hunt down those compromised civilian cells out in the tissues. But wait, how do our infantry troops actually know who's friend or foe when they reach the battlefield? Well, they use MHC molecules. MHC stands for Major Histocompatibility Complex, and they act exactly like molecular display cases or ID badges on the surface of your cells. You've got MHC class 1, which is found on almost all normal cells in your body, and then there's MHC class 2, which is a special badge reserved exclusively for professional immune cells, like our dendritic spy or B cells. These display cases hold up those tiny epitopes we talked about earlier for inspection. This creates a perfect lock-and-key system governing our rules of engagement. CD8 killer T cells scan the MHCI badges. So if a regular lung cell is infected, it displays the viral fingerprint, and the T cell docks to confirm the cell is compromised. On the other hand, CD4 helper T cells read the special MHCI2 badges on our immune antigen-presenting cells to receive their orders and coordinate the broader war effort. It's a completely foolproof system designed to ensure our troops don't accidentally attach healthy tissue. Now, when a CD8 killer T cell actually finds an infected target, it doesn't just smash the cell to pieces. That would just release the replicating viruses everywhere. Instead, it executes a precise, 3-step breach and clear maneuver to neutralize the threat safely. First, it uses a protein called perforin. Next, it deploys a payload called granzyme B. And finally, this payload activates a cascade of internal enzymes called caspases, which basically force the cell to quietly dismantle itself. During the attack, the killer T cell literally fires molecules of perforin that assemble into a cylindrical tube, blowing a physical hole, a lytic pore straight through the enemy's cell membrane. Once that breach is made, it drops granzyme B right through the pore like a microscopic, explosive charge directly into the cytoplasm. The infected cell has absolutely no defense against this kind of targeted delivery. The aftermath of this strike is incredibly clinical. The cytotoxic T lymphocyte docks, delivers the fatal blow, and then just cleanly detaches to go hunt its next target. The infected cell is then forced into apoptosis. And this isn't some messy explosion, right? It's a highly controlled self-destruct sequence that neatly packages the viral particles into digestible fragments, entirely preventing the virus from spreading. It is just ruthlessly efficient. But remember, we're talking about an evolutionary arms race here. The invaders absolutely fight back with incredible counter-espionage tactics. Because if viruses know their survival depends on staying hidden from the CD8 T cells, they're going to attempt to jam our communication lines. They deploy specific proteins to sabotage those MHC1 display cases, making sure that the infected cells remain totally invisible to our T cell patrols. Because if the T cell can't see the viral fingerprint, it doesn't know the cell is infected, and the virus gets to just replicate in peace. And this is where we see the true, albeit terrifying, genius of viral warfare. Sneaky viruses like HIV, cytomegalovirus, and adenovirus will actually intercept our defenses at multiple stages. They produce proteins that sabotage the TAP transporters, which stops the viral fragments from ever entering the endoplasmic reticulum. Other viral proteins, like HIV's NEF, will literally force the cell to pull the MHC1 ID badges back inside before they ever even reach the cell surface. It is a masterclass in molecular sabotage. So, with millions of these microscopic battles happening simultaneously and viruses constantly trying to turn invisible, it raises a really fascinating question. How in the world do our scientists and doctors monitor this hidden war zone? If the cells are microscopic, how do we count the troops, analyze the special forces, and actually figure out if a patient is winning or losing the fight against an infection or an immune disorder? The answer is a piece of absolutely breathtaking technology called flow cytometry. Think of this as advanced surveillance for doctors. We can take a sample of blood or tissue, suspend the cells in a fluid, and stream them single file past a series of lasers at speeds of up to thousands of cells per second. As the laser hits each individual cell, the light scatters. And by measuring that scattered light, we can count cell populations, sort them by size, and even detect specific biomarkers to see exactly how many CD4 or CD8 T cells are active in a patient's body in real time. And by tracking these cellular battlegrounds, we can actually intervene. This is exactly how we develop vaccines that pre-arm our humoral response. See, if we expose the body to a harmless epitope beforehand, our B cells go ahead and create neutralizing antibodies. Then, when the real virus finally shows up, these Y-shaped antibodies swarm it, essentially intercepting the enemy before they can even breach the castle walls. The elegance, the coordination, and the sheer lethality of your adaptive immune system is truly a marvel of biology. But as you reflect on the brilliance of your body's defenses, I want you to consider this. Viruses mutate rapidly. They are constantly probing for weaknesses in our armor and developing brand new counter-espionage tactics. In this endless, evolving war against microscopic invaders, the battlefield literally never rests. So, what new defense strategies is your body adapting and developing inside of you right at this very moment?